So, hello everybody. Today we will discuss about a very important pathway that is NF-kappa beta pathway. Now, this pathway is involved in a lot of things, right? Maybe an immune response, T cell activation, apoptosis, mechanotransduction, cell proliferation, a lot of pathways, right? Now, it is pretty simple, but there are so many complexes, or the names are pretty complicated. It seems a bit difficult. So let's decode bit by bit. Now, this NF kappa thing, this NF kappa beta, it either function as a homodimer or a heterodimer, and it is formed by the members of REL family of DNA binding proteins, right? Now, what these REL families are, components are, which is REL A, C REL, REL B, REL 105, and this, right? Now, these are the proteins basically. Now, this P105, if I talk about the precursor of P50, so basically it's being broken down into P53, then later on, as being a homo or a heterodimer, this P50 will later on bind with rel B to form a complex, right? Now, why we are considering to these group of protein? Because it has a rel homology domain, so therefore it includes DNA binding domain, dimerization domain, and nuclear localization signals. Now, why it's pretty important here is that this nuclear localization signal is bounded by I kappa B proteins. So, into an inactive form, right? So, NF kappa beta remains inactive in the cytoplasm because it is bound by this protein. So it includes this alpha, beta, gamma, BCL3 types of protein which keep the complex in an inactive state. Now, the activation. So you need a kinase. Let's phosphorylate and let's break it down. So there is an I kappa beta kinase complex. So if it is alpha, it is basically involved in non-canonical pathway, basically the other functionalist. Major, it's not just a major pathway, it's just another pathway, right? Not the typical function. So this is I kappa beta. It is a major kinase in the canonical pathway, and this is very important. IK gamma. Now, why? Because it is a regulatory scaffold subunit which is required for canonical stem. So it's basically a scaffold. So it will keep the complex in a bounded form. Now we will discuss about how signaling occurs. Let's join into the major thing that we want to discuss. Right? This beautiful thing that we have. Now you get a signal. Now what these signals are? This is TNF alpha. There is a receptor. It can be interleukin 1. It can be pathogen associated molecules or any cellular states. So what will go? Now this is an inactive complex bounded with the protein that is hiding their NLS, right? So with the help of kinase, I will remove this inactive protein. So with the help of ubiquitination, degradation of phosphorylated I kappa beta in proteasome, you have a free NF kappa beta protein which will have rel proteins, and they have those binding domains which can easily bind in there, right? It will recruit certain cofactor proteins due to NLS being free, it can go into the nucleus, right? And there is transcription of target genes. So this is the overall thing you can see, right? Now, first IKB is phosphorylated and later on IKK phosphorylates first this complex. Now the thing is mechanism of non-canonical, it's pretty similar only. It just the thing is that it is first processed into this or P50, right? And therefore there is this heterodimers being formed, and then whole complex will translocate to the nucleus. So the mechanism is pretty simple. Just there is a processing time in between. Now I have to discuss about the roles and functions. So you can see here chemokines, inflammatory response, angiogenesis, adhesion, metastasis, like so many roles are there. Anti-apoptotic pathways, dendritic cell maturation, T cells, macrophages, a lot of functions, right? Now let's say the temporal dynamics. So when this is synthesized, it binds to this, it permanently dampens the response within R. So means if I have a TNF pulse, then the activity would be strikingly increased. But if there is a continuous TNF exposure, TNF is this thing, okay? Is exposure, so there is a sustained response. It will not drop like this. There will be a sustained response because it's like I need to protect myself, right? Now, this sustained response forces this NF-kappa B levels into oxalations due to mechanical time delay between the degradation and new synthesis, right? Because in each pathway, your first the protein will be degraded, right? This will be degraded and the complex will be going into the nucleus. Also, you want the other components to be there also. So you need to synthesize them also to form the pathway, right? So there is a time lag, and therefore the amount of this nuclear NF kappa beta dampens or moves like an oxidatory pathway, right? Right, short pulses will only activate the genes which are in first response. And if there are a lot of signals, then late responding genes are activated. So that's another thing. Now, the other point that we have to discuss is that this is basically a negative feedback loop, right? That if there is too much of this, then there will be a lot of cell proliferation, right? So to stop this, we need even IKB synthesis so that I can inactivate it, right? Now, this regulates both cell survival as well as apoptosis, and it depends on the cellular context. We'll see how. Now, important is that it's not just that you are immunology based signals, but it can also be activated with the help of mechanical signals like shear stress, pulling or pushing sort of stress, right? The modulators for them are just say like integrins, PAC, Rho, actinomycin, microtubules. So these are basically the mechanosensitive regulators. Now we'll discuss a very important thing. This is very important in terms of which cell can either promote cell survival or cell death. So in anti-apoptotic survivals, basically it promotes beta cell activation, hepatocytes, T cells, right? Protection from apoptosis. And many viruses exploit this system so that they can live, right? Stopping from apoptosis means being live, right? Apoptosis is nothing, cell death, right? Now, now it can contribute to apoptosis under certain condition if there is a fax mediated response. So it is highly context dependent. Now another, so you can just see, this is the portion which I covered. Now this is the canonical versus non-canonical. So the thing is that First, the precursor is degraded, and the complex is this. Here it can be rel C-rel P50. Here it is P52 and rel B. It unmarks NLS and it produces active P52 from precursor. So there is a slight difference, okay, right? Another post-translational modification and site-specific phosphorylation. So basically, they basically phosphorylate at serine residues in general in NLS, NF-kappa beta based signaling pathway. Right? I hope nothing is left as such. Yeah, this myocardium stuff, right? So myocardium is something which is very important in terms of muscle differentiation. And this high myocardium blocks NF-kappa beta DNA binding. So it will reduce your inflammatory gene expression. So it's just that myocardium and NF-kappa beta B are antagonistic. Thank you. I hope I have covered every aspect that I could. Yeah, these are the sources that I have used along with Albert's. Thank you.